Hello and welcome to my presentation about my project on predicting optimal combinations of perturbations for generalized cell-state conversions using combinatorial optimization and machine learning algorithms. My name is Shaurya Agrawal and I'm a high schooler at Adlai Stevenson High School, and this project was done at the MIT Kellis Lab under the direction of Dr. Martin Woolwend and Professor Manolis Kellis. So first, I would like to talk a little bit about the transcriptome. A cell's transcriptome is defined by the expression of its genes or the mRNA profile of that specific cell, and it's crucial to defining cellular functions and processes such as cell identity as shown in this diagram. So now this begs the question, what happens if you change the gene expression levels or the expression of the transcriptome? Well, that causes changes in the cellular functions and processes of that cell as well. Now, this begs the question, what if we induce changes in the transcriptome? So, what if we make planned changes in the gene expression levels? Well, that would lead to specific choosing for specific phenotypic effects. And this is known as cellular reprogramming, where the transcriptome can be reprogrammed for various phenotypic effects. And cellular reprogramming offers immense promise in regenerative modeling, medicine, disease modeling, disease reversion, and various other areas of research that are crucial. And how is this done? Well, it's done by manipulating gene expression as shown in this diagram. Now, how do we manipulate gene expression? Well, we use something known as transcription factors. Transcription factors are great targets for manipulating gene expression in cellular reprogramming. This is because transcription factors are a regulatory gene that bind to promoter regions of the specific gene to be expressed, and they can either repress, enhance, or perform cofactor enhancement with other transcription factors as well. And since transcription factors are sort of a paired gene where they can manage the gene expression levels for various different genes, um, changes in specific in a few transcription factors can lead to large downstream effects such as complete change in cell identity itself. So this makes it a great target for manipulating gene expression and cellular reprogramming in translational applications. So now what's the issue? Why has cellular reprogramming not been fully realized yet? Well, the issue is that due to cost, most research currently focuses on stem cell differentiation. So stem cell differentiation is sort of a simpler task in cellular reprogramming that tries to figure out how to turn stem cells into specific cell identities. The research hasn't been able to do experimental research or projects on more complex tasks such as disease reversion, so disease to healthy cell state reversion, or transdifferentiation, which is going directly from one cell type to another cell type, skipping the stem cell stage in between. And so another issue is that due to the high cost of experimental projects, the usage of in silico projects are also lacking. The usage of in silico and transcriptome wide experiment simulation models have been very limited, and this has led to no efficient or generalized models for cellular reprogramming, especially for converting disease cell states into healthy cell states or transdifferentiation. So, what is the solution? Well, the solution is to develop a model that can predict the optimal transcription factor perturbations to induce on a specific cell in order to go from a disease cell state to a healthy cell state, as well as perform transdifferentiation using efficient in silico models that have high generalizability. And the way this will be done and the data that will be used is perturb-seq data. So, how was the data acquired? Well, the data was acquired from a large scale study and the paper Replogle et al. And this data contained two specific data sets, or two cell lines especially. It contained genome wide leukemia cell line, which is or erythroleukemia cell line, and retinal pigment epithelial cell line, which is a healthy eye cell. And each data set had five controlled non perturbed cells as well to do the comparisons with. So essentially, how this perturb-seq works is you have a control cell line. In this case, it was the erythroleukemia cancer control cell line, and in that cell line, each gene is perturbed. In this case, a 30% knockdown on the gene, and then after the perturbation, the gene expression levels of all the genes are measured again. And this is done for all the 9,854 genes in the model or in the data set. And this gives us three data sets. One is the cancer control, the original cancer control. The other is the results of all the perturbations done on the entire and on the entire transcriptome sequenced, and the other is a healthy control cell line. Because remember, our goal is to go from a cancer control cell line to a healthy control cell line and computationally simulate that. So the data preprocessing, essentially what it showed us was that we got 550 transcription factors and 7,094 genes in the transcriptome. And this is a much larger scale study than other research projects done before, or research done before. Essentially, these research studies focused on specific pathways. So that meant focusing on only specific transcription factors or specific genes in the transcriptome, and not a wide scale study with hundreds of different transcription factors and thousands of genes sequenced like this study. So, what were the methods? After the data collection was done, we did a benchmarking analysis because we needed a basis for validation, since the novelty of our research meant that there weren't direct models for our models to be compared with. So, we used past research and statistical analyses to develop our own benchmarks in order to do comparisons. And essentially, what these models did, which we had three: vector addition, greedy algorithm, constrained OLS, and mixed integer linear programming. They tried to figure out the optimal perturbations to be made from the 550 transcription factor perturbations that we had available to go from to do the most optimal conversion from the initial erythroleukemia cell state to the target cell state. And in the vector addition greedy algorithm, essentially how it worked was that it converted the cell states all into vectors and tried to figure the optimal addition vectors to get the optimal similarity between the computed vector and the target state vector by adding all those addition vectors, or better yet, the transcription factor perturbations. So the benchmarking methods, there were two benchmarking methods, and after both of the benchmarking methods were done, the top five transcription factors were extracted. And essentially, the goal of these benchmarking methods was to find single transcription factor perturbations that would be most useful rather than combination transcription factor perturbations. Because this is essentially what past research has done, is using statistical methods to figure out single transcription factor perturbations rather than combinations, which has been lacking. So after doing this sort of analysis, we sort of gained our results. So this is the heat map of the entire data set present. And here are the results of the two benchmarks. So you can see in the top row, we have the epithelial control for both. And in here, we have the cancer control. And all of the other rows are single perturbations made. And so you can see how there's essentially no difference between the cancer and the perturbed single transcription factors, and the epithelial control differs completely, or a whole lot. It varies a large amount from the rest of the single transcription factor perturbations, which goes to show that single transcription factor perturbations are not very useful for these types of complex cell state conversion tasks. So let's quantify that. In terms of the cosine similarity score, the maximum that any transcription factor perturbation got, which was the GATA 1, was 0.64. And out of those 0.64, we have we have two genes, which was ETB4 and zinc fingers, and both of these are well-known cancer or oncogene pathways, and but these two, which was another zinc finger and GATA1, are well-known cellular differentiation pathways. So after doing these analyses and doing running this model, we found a combined vector of 9 transcription factors. And as you can see in the results here, the combined vector from the model beat out the other results significantly with a combined vector score of 0.90. And this model was run on the top 75 DEGs. The reason it was run on those top 75 DEGs was in order to also further translation applicability, because in terms of translation applications, it isn't very feasible to try to convert all expression levels of all the genes, especially ones that might not be very effective or might be non-coding, might be not very useful for this type of analysis. So we extracted the top 75 DEGs to do this analysis on. And here you can see that we have another score that we use. So other than the cosine similarity, we had a projection score, which takes into account both direction and magnitude, which can be very useful for this problem. And the cumulative scores show that the first few transcription factors added the most to the model. So GATA1, all these top transcription factors in cellular differentiation and oncogene pathways added the most. And in terms of DEGs, the cosine similarity score is definitely much more reliable than the projection score, as it doesn't vary as much with the DEGs varying. So the results of the constrained OLS regression, which rather than just predicting binary perturbations, also predict titrations of perturbations, was actually surprisingly um, a lower conversion rate than the greedy algorithm, though it still extracted crucial results. And the reason it was a lower conversion rate was one reason was because the the error used, the objective function is the error rather than the cosine similarity. So it's the Euclidean distance rather than cosine similarity or projection score. So we needed a similar gradient based regressor with a modified objective function. For that, we used an optimizer nonlinear programming SQP solver, and it gave us very similar results, and again, surprisingly less than the greedy algorithm. So the literature review shows that the model was able to find both cell differentiation associated TFs and oncogene pathways associated TFs, and top TFs such as ETV4, GATA1 were first choices in every randomized run of the greedy algorithm. For conclusion, future work, essentially what we found was that this combinatorial optimization model, along with machine learning models, were significantly increased similarity with epithelial vector when we did combinations of perturbations, transcription factor perturbations, providing novel results for cell state conversions and simulations. In the future, we would like to use large amounts of TFs, even though we already did use large amounts of TFs in order to better our results. Thank you.